Hi, everyone. Welcome to the third Burst General Meeting. I'm so glad you guys are here. We have a great speaker for you guys today. And we have a lot of cool announcements, and our ID is coming up this weekend. So very excited about that. All right, we're going to go ahead and get started. But if you're having any trouble, or if you know someone having trouble getting into the general meeting, feel free to email Tiffany or try to find the general meeting links from the past two meetings. All right, so we're going to start off with an icebreaker. It is October now, so Halloween is coming up. What are you guys going to dress up for as Halloween? Feel free to unmute yourself and share or put it in the chat. Medusa. Tuba, are you dressing up as anything this year?
I don't know yet. We'll see. We'll see when Halloween comes around.
Okay. Jake, you said SpongeBob. Do you already have that costume lined up? It's a onesie. Okay. Very nice. That's easy. If you guys didn't know, the Livingstones actually give out full-size chocolate bars on Halloween. I don't know if they're going to do it this year because of COVID and everything, but if they do, you guys should go by. All right, so here are all the officers. If you haven't had a chance to meet us, please do send us an email, or if you see us on campus somewhere, just stop us and say hi. We would love to talk to you and get to know you more. And then our member of the bi-week is Andrew Munoz. He is a editor on Scientia. He's also a biology journal club leader, and he's just been doing great work these past 2 weeks as a burst member. So if you see him, please congratulate him or go to one of his biology journal clubs. Andrew, are you here today? I don't think he is, but he gets a free t-shirt.
Okay, so just a quick reminder that you need to earn a minimum of 16 points by the end of the semester in order to remain an active burst member. And those 16 points can be further divided into 4 different categories. So a minimum of 7 general event points, 4 journal club and service hours, and 1 special event. And so the only event that falls under the last category is Research Internship Day, which is this upcoming Saturday. So we look forward to seeing everyone there. And also, if you're a part of a research lab this semester, you're welcome to fill out the form links below to earn 6 points towards your research credit. And just on the basis of points and participation, and this really applies to those who haven't really attended any events outside of these general meetings, just make sure you're maximizing on those opportunities. And that's kind of a nice segue into the next slide. With our weekly email. So these go out every Sunday night, and they should be your go-to resource for upcoming burst events, as well as just other events hosted by other pre-health organizations that would be equally beneficial for everyone. So make sure you're taking the time to read those. And also there's just a meme, a really great meme, if I do say so myself, at the very end. So that will be worth your time somewhat.
And don't forget, you guys, to pay your dues. If you're a new member, it's $25. And make sure you go ahead and fill out the new member form on the website. If you don't see your ID number on the points record, it's probably because you haven't filled out the new member form. So make sure you've done that. And then if you're a returning member, it's $15. And you can pay your dues on PayPal this semester. We're just going to do everything electronically. And we've actually ordered our shirts, so they should be in about 2 weeks, and then we'll get those to you guys.
Hi everyone. So as you all know, RID is this Saturday, October 10th. So like Tiffany mentioned, this is a required event for all burst members to get that one special events point for the semester. It's going to be fully virtual this year, and all Zoom links will be posted on the Baylor burst website. Sorry. And please join the welcome Zoom call at 1:20. I know the event says that it starts at 1:30, but please join at 1:20 so that you can get some instructions on how to get that attendance credit and stuff like that. You don't have to dress up since it's a virtual event, but please be presentable and professional in all of your Zoom calls. All right. And our keynote speaker for this year will be Dr. Maria Batasi, who is the co-director of Texas Children's Hospital Center for Vaccine Development. She's also the associate dean and professor at the National School of Tropical Medicine at the Baylor College of Medicine. So come learn about her research and also hear her talk about global health. It's going to be a fun time. All right, so I'll give you some overviews about the attendance instructions, and I'll also go over this the day of RID in case anyone's confused. But to receive full credit for RID, you need to attend 3 poster presentations, each from a different discipline, and then you need to attend 2 breakout sessions and 1 keynote speech. So an easy way to remember this would just be 3 and then 2 and 1.
Hi guys! For upcoming journal clubs, there's still room in our physics journal club tomorrow, so feel free to sign up. We have epidemiology, research ethics, neuroscience coming up this next week. The week afterwards, we have a phage journal club. You do not have to be in Dr. Adair's Bears in the Sea class to participate in this journal club. And we also have a Mer Physics Club.
So our post-meeting social today is going to be Among Us. So that'd be fun to play. Join us right after this meeting. Same Zoom link and all, just stay on it. And we can make and break some relationships. Then burst family groups, those are coming up. Everyone should have been contacted and allocated a leader by now. Check your emails, check the junk emails, check your group meets. If by any chance you haven't and you checked all of that, then just message me or email me at this email right here, or just message me right now and I'll look into that and get back to you as soon as possible. And then we have a burst scavenger hunt coming up on October 21st, 6 to 7. This is tentative, but this will be a family group versus. So all the more reason to make sure you're in a family group. And if you're not in one or you chose not to be in one, you can still attend and we'll just put you with a certain group. And then the winning family group or the winning group gets a surprise.
Okay, guys, so scientific 21 submissions are now open. So if you're interested, it's open to all Baylor undergraduate researchers, and you can publish review papers, original research, or abstracts. And if you want to learn more about submission, or if you know a friend who wants to submit, you can show them our website. Okay, and then the scientific committee has been working hard on our budget research newsletter. We have a lot of cool information in it. We have information on upcoming internship programs, both virtual and in person. We have an interview with BCM's Dr. El Sali about the Moderna COVID vaccine. And we have a lot of testimonials and interviews from really cool Baylor student researchers. So it's new released on November 2nd, and you'll get an email about that, so be sure to check that out.
All right, hey everyone. So we're gonna have a few upcoming workshops. Actually, not a few, a lot. So this coming Monday at 4:30 p.m., we're gonna be hosting the Lab Techniques workshop. So this one will be really interactive, and you'll be able to ask questions throughout this workshop. And the goal of this workshop is to teach you all the basics for succeeding in your upcoming labs, such as biology or chemistry or even physics, and even for your future pursuit for research labs. And so we'll teach you things such as pipettes, how to use centrifuges, or how to use gel electrophoresis, instruments like that. And so we'll teach you the instructions for those and the nitty-gritty details for that. So if you're interested, we'll be sending out the Zoom link once again in our upcoming website and our emails. Actually, it's already including the emails, so you're good if you want to check it out. And another workshop we have is the types of research and the MCAT prep and MD versus PhD. And so in November, we'll also be including a LinkedIn workshop and a bioinformatics seminar and the GRE workshop. And so all of the Zoom links for these will also be included in our weekly Sunday emails.
All right, now it's time for our general meeting speaker. Today we'll be hearing from Dr. Mary Lauren Benton. She is an assistant professor of bioinformatics here at Baylor University. She received her PhD from Vanderbilt University in biomedical informatics, and she was actually a Baylor Bear and graduated with a BS in bioinformatics. So please join me in welcoming Dr. Benton.
Hi, thank you all for that introduction. And yes, I was a former bioinformatics grad, so I'm excited to be back in Waco and excited to be sharing my research with you today. All right, so I'll just go ahead and get started. And today what I want to talk to you about is what I've titled Identification, Interpretation, and Evolution of Gene Regulatory Enhancer Landscapes. And we'll dive into what all of that means here in just a couple of slides. But I want to start out by motivating why studying genetics is so important. And to do that, I want to highlight first that many common and extremely impactful diseases have a genetic component or genetic risk factors. For example, this graph is the top 10 leading causes of death in the United States from 2018. And the majority of these, now highlighted in red, either have genetic components or genetic risk factors. And with the rise of direct-to-consumer testing like 23andMe, and the growth of precision medicine through things like the All of Us initiative, it's becoming increasingly important to both healthcare providers and to the public to understand how genetics play into disease risk. However, genetics is difficult to study because simply the human genome is complicated. The pictures here are some of my favorites. They show books containing a printout of the first human genome sequence in really, really tiny print. And the fun fact of the evening is that if a person were to type out the human genome at a relatively quick rate for 8 hours a day, every day, it would take more than 50 years to complete the job. So understanding how this sequence encodes instructions for the proper functioning of every cell in our bodies is a huge undertaking and one that requires the use of computational techniques. Furthermore, the books or DNA sequences for each person all look a little bit different. DNA sequences have millions of small differences. There are an estimated 4 to 5 million of these single nucleotide changes between any two people's genomes, and many more in populations around the world. These genetic differences or variants can influence traits, kind of like how I have blue eyes or some people have red hair, but they can also influence traits in terms of disease risk. Before we think about the impact of these genetic differences, I want to take a step back though to our human genome from earlier. When I say genome, maybe not for this audience, but typically the first image that comes to mind is a double helix that's floating somewhere in space. It's probably blue. On the right is actually a transmission electron microscopy image of a DNA fiber from 2012. And if you look at the red arrows in that zoomed in portion at the bottom, you might be able to make out some of this helix structure, although it's definitely not blue. However, we can also envision the genome in a schematic form more like this. So the DNA sequence exists as a helix, which is wrapped around complexes of histone proteins. It's packaged into chromatin, and ultimately this forms the chromosomes. But for now, let's picture the genome just as a simple sequence of letters. In the schematic, part of the genome includes genes that I've highlighted in yellow. Genes code for proteins that provide structure and perform essential processes in the cell. And the DNA of the genes is transcribed into RNA, which is then translated into protein. If we were to add in the arrow for DNA replication, I'm sure all of you would recognize this process as the central dogma of biology. But what's important to see in the schematic is that protein coding genes actually make up an incredibly small fraction of our genome. So what about the rest of the genome that I've now highlighted here in red? It does have function, since previous work has shown most of the genetic variants I spoke about a few slides ago are actually located in this part of the genome. In fact, less than 10% of the genetic changes associated with traits affect genes. The vast majority fall into what I will refer to as non-coding regions, meaning they don't affect the sequence or the structure of proteins. So what do these regions actually do? Well, some parts are responsible for turning genes on and off. In our schematic, I've highlighted some of these in green, and I'll refer to these as gene regulatory elements. You can think of them kind of like switches or dials that control which genes are transcribed at a given time in a given cell. And despite the fact that all the cells in our bodies have the same DNA sequence, they actually express different genes at different levels. By express, I mean that the gene sequences are transcribed into RNA at different levels. And this is because the genes are regulated by different gene regulatory elements with cell type specific functions. Using this heat map, I'm showing you 6 different tissues along the x-axis, liver, muscle, lung, and 3 brain regions. And the expression level of different genes is shown on the y-axis, using the light to dark green color to represent the level of gene expression. If we look at this plot, we noticed a few trends. One is that expression is similar in similar tissue types. So the patterns for our 3 brain regions all look alike. We'll also notice that some genes are expressed across all cell types or are broadly expressed, while some genes are expressed more specifically in a single cell type, such as this cluster of genes expressed primarily in the liver. Understanding how genes are regulated is important both to answer questions about how the genome works and to better understand the health implications of genetic variation. And we need tools in computer science and bioinformatics to make progress in this area. So in this talk, I'm going to focus on 2 ways that I've used better methods to improve the status quo and how biologists interpret and study gene regulation. First, I'll talk about identifying and studying gene regulatory elements on a genome-wide scale. And second, I'll talk briefly about how I've been integrating available genomic data sets to help us move beyond considering regulatory elements in isolation, and instead enabling us to better understand how they function in a broader genomic context. So we'll start with our first question. For this part, I'm going to talk about a specific kind of regulatory element called an enhancer. This is an example of an enhancer in red that turns on a gene in blue called sonic hedgehog. And this gene is involved in limb development. So you can see in this picture, the red arrow shows the activity of the enhancer in the developing limb bud of the mouse. And we'll remember our enhancers are really just short DNA sequences that are regulating the expression of this gene. And what's interesting here is that even small genetic mutations or variants can change the way the gene is expressed. In this case, even single base pair genetic variants can result in something called preaxial polydactyly in humans, or this extra fingers. So we looked at a single example of an enhancer, but it actually remains difficult to identify them more broadly. So these enhancers are cell type specific sequences of DNA that regulate the transcription of genes by binding to specific proteins called transcription factors. And currently, we actually use a range of more easily measured attributes that are correlated with enhancer activity in order to map these enhancers on a genome-wide scale. So let me orient you to this schematic a little bit. So our gene here is shown on the far left side, and the line down the center of your screen is the actual DNA sequence. You can see that's wrapped around some of these histone proteins that are the gray cylinders. We also have examples of transcription factors that are binding to specific sequences. And the patterns that, or motifs that the transcription factors bind in the DNA sequence are shown by the different colored blocks. So when identifying enhancers, we often look for attributes like places where DNA sequences are available for binding. We sometimes call this chromatin accessibility. We also look for specific patterns of histone modifications or the chemical groups added to histone proteins that occur in specific combinations. We can look for the binding of transcription factor proteins, or we can look for the short sequence motifs in the DNA that they bind. And we also know that some enhancers are actually transcribed themselves, and they are transcribed into a special kind of RNA called enhancer RNA. And we can also look for that. And these attributes broadly are used either alone or in combination to try to identify enhancer regions. So your first question might be, does it matter that we have so many different ways of identifying the exact same thing? But my work has showed that when interpreting non-coding variants of interest, it can matter a great deal. So as an example, let's say we want to look at genetic variants associated with a risk for heart disease, and we found dozens of potential candidates, a few of which are highlighted here as the small vertical bars at the bottom of the screen. If we overlay, we can see that none of these variants actually alter a gene. So one hypothesis is that these mutations increase risk of disease by disrupting regulatory activity. So if we overlay known maps of enhancer regions, we can try to identify potentially disrupted regions. And in this case, we might use one of the many enhancer definitions I described on the previous slide. If we were to use the green map, then we would prioritize this particular variant for experimental follow-up, which costs time and money. But if we were to choose the blue map of enhancers, then we would prioritize a different variant. And if we were to choose the pink map, we might conclude that there's little evidence that these variants disrupt an enhancer element and go looking for our mechanism somewhere else. In reality, each of these candidate variants in the example were tested, and only the two highlighted here in yellow were actually causal. These two variants do contribute to the risk of heart disease by disrupting gene regulation, but they weren't predicted by any of the definitions shown here. Because the prioritization of genetic variants can change based on the enhancer set, this can result in both false negatives and false positives. This is a larger problem because most studies consider only a single approach when defining enhancers. And so we hypothesized that differences between enhancer identification strategies would have an influence on the set that they generated and ultimately on downstream biological interpretation. For example, if you chose the enhancer definition shown in blue, you would end up with one set of downstream results. But if you chose the enhancer set in pink, you might end up with a completely different set of conclusions at the end. So understanding the similarities and differences between strategies in current use is really essential, both to assess how stable our conclusions are and to improve our understanding of gene regulatory architecture. So we compared genome-wide enhancer sets identified by 11 diverse representative assays and computational techniques. And so each of these that are shown on the left have been used in a high-profile published paper over the past few years. They use a range of different input features, so looking for things like histone modifications or those chemical groups added to the histone proteins. They looked for the binding of specific transcription factors. They looked for chromatin accessibility, or regions of the sequence where transcription factors are able to bind, and looked for things like enhancer RNA. Using all of these input features, these definitions combined the experimental data using a range of computational processing techniques, including machine learning in both supervised and unsupervised frameworks. So we asked our first big question, which is really, do enhancer sets actually agree with each other? So this schematic is showing two enhancer sets, one A and the other in B, where the enhancers are shown by the gray blocks. And in order to determine whether enhancer sets predict the same location, identification methods predict the same locations to be enhancers, we used a technique called the Jaccard Similarity Index, which is looking at the intersection, or the places where these two approaches agree, over the union of all of the sequences that were predicted to be an enhancer by either approach. This gives us a score from 0 to 1, which I've plotted on this heat map here. So this heat map shows the Jaccard similarities between all pairs of enhancer sets in the liver. And we can see that all of the similarities are very low. Most of them are below 0.3, which means that while each method predicts enhancer elements, they're actually identifying very different genomic locations. If we were to look at a cell line, so maybe K562, which is a cell line, a cancer cell line, where we have additional approaches that we can compare, we still see that the overall similarity is extremely low, much lower than we would expect, considering that all these approaches are trying to identify the exact same type of regulatory element. And this matters because if you remember the heart disease example from the beginning, the dissimilarity that we just saw in the previous two slides means that the situation we described here is actually a very common one. I quantified how often this happens on a genome-wide scale, represented here as a stacked bar chart. So for each cell type, starting with our cancer cell line, I calculated the proportion of genetic variants that influence disease that are overlapping enhancer regions. And we can see by this, with this big green bar, that the majority of these trait-associated variants are identified by only a single method, almost 70%. And this trend generalizes across many different cell lines and tissue types. And actually, none of the variants that I considered here are actually predicted by all of the methods. So this suggests that a simple variant prioritization and interpretation of variant function is often contingent or dependent on the enhancer set that you use. So we could largely ignore this problem if we knew which enhancer set was the best. But unfortunately, there isn't any kind of gold standard or right answer. So we have to use other proxies. So one idea is to use expression-associated genetic variants, sometimes called EQTL. An EQTL is a variant where the allele that you have at a given position, or the genotype that you have in a given position, is predictive of the level of gene expression of a nearby gene. And we expect that real enhancers should have these variants. In fact, we expect that real enhancers should be enriched or have more of these variants than other places in the genome. And so we might expect the very best method at predicting enhancers to be the one that's most uniformly enriched for these variants. To test for that, I developed an enrichment framework that uses permutation to look for the amount of overlap between enhancers and variants. So here we have our enhancer set E and our variant set V. And we can count the number of regions that overlap. So the number of variants that overlap an enhancer in our observed enhancer set. Then we can take our enhancers and actually randomly shuffle them throughout the genome and recalculate the amount of overlap between our new random set of enhancers and the genetic variants. And if we do this a large number of times, we can actually create a distribution of how much overlap we would expect with random regions in the genome. And if we compare the amount of overlap that we observe with what we expect based on this random distribution, we can calculate a p-value for the observation by comparing it to this expected distribution. And so our p-value tells us what the probability of obtaining the amount of overlap that we observed, or something more extreme, given the null distribution that we calculated using our random shuffling approach. And we use this permutation framework to calculate enrichment for each enhancer set across multiple cell types. So in this plot, the x-axis represents each of our enhancer identification methods, and the y-axis represents the enrichment, or how much more overlap with expression-associated variants that we observe compared to what we would expect based on that random distribution. Here, the colors of each point represent the 4 different cell types that we were looking at, 2 cell lines and 2 primary tissue samples. We can see here, while there is some variability in the amount of enrichment, no method stands out as a clear winner for the highest enrichment across all cell types. And if we remember our Jaccard similarity analyses from earlier, we would know that actually most of these are very different enhancer locations. So while the enrichment is the same, these are actually very different genomic locations. So maybe instead of finding the best method, we can try a new approach. Because while there is a lot of disagreement, in some places there's actually agreement. And so we subset enhancers in our group based on how many methods predicted that region to have enhancer activity. So we started with those that are unique to a single method and ended with those that are shared across all of the methods, and asked whether there are different underlying sequence patterns between shared and unique enhancers. Essentially asking, is there something distinctive about enhancers that are reproducible or more highly shared across methods? We set this up as a machine learning problem, or a prediction problem, to look for properties that distinguish the 2 classes. So using sequences of shared examples as our positive set, and sequences from unique enhancers as our negative set, we can train a machine learning model to actually predict the classification of a new enhancer sequence. So if we were to add in new data or sequences of unknown enhancers, we could try to predict whether these enhancers are shared or unique. From here, we can look at the importance of the features in our machine learning model to try to learn something about what sequences distinguish these 2 enhancer classes. So here, we use sequences of shared enhancers, like I said, as our positive set, and sequences of unique enhancers as our negative set. And we decided to distinguish enhancers using short sequences, or K-Mers. We chose K-Mers because they actually represent a really useful biological feature, like transcription factor binding. And so from here, to distinguish enhancers using these K-Mers, we first created a K-Mer Spectrum, which is essentially all of the short sequence motifs of a specific size, here of size 5, across the entire enhancer sequence. We can calculate each K-Mer and the frequency that with which it appears in each enhancer sequence. And then use what we call a K-Mer Spectrum kernel in a support vector machine, which is really just an algorithm, using the frequency of these features across different enhancers. So our matrix of features, input features here, is one row for each enhancer, and the observed frequency of each K-Mer in that enhancer sequence. And our support vector machine, or algorithm here, is trying to classify sequences as either shared or unique. And what an SVM model does is really, it represents all of the examples that we put in, so all of our enhancer sequences as points in space. And it tries to find or separate the 2 categories, that are positives and negatives, by a gap that's as wide as possible. So in machine learning terms, we call it a hyperplane that maximizes the separation between these 2 classes. And so here our hyperplane might be drawn down the center, and we want to maximize that distance between the shared and unique enhancers. And SVMs use these different kernels, like the K-Mer kernel that we talked about earlier, in order to map our inputs from a high dimensional or really large feature space into a space where we can actually separate them with a single line. So after training this model, we can see how well the model performs and ask whether there are sequence differences. And so before I show you the accuracy of this model, let me just orient you to my plot a little bit. So on the x-axis here is our false positive rate. The y-axis is our true positive rate. And if we were to create random predictions, we would expect what we call an area under the curve of about 0.5. So we expect a line that runs diagonally across this plot with an area under the curve of 0.5. If we were to train a model that made really good predictions, we would expect our line, our accuracy to look more like this, and we would have an area under the curve somewhere closer to 1. So when we plot our model performance on the same plot, we get an area under the curve of 0.6. That means that our K-Mers don't actually distinguish very well between unique and shared enhancers. Our model does not perform very well. And so this actually suggests that our sequence patterns are not very predictive of reproducibility. So given that the model trained on our DNA sequences did not perform very well, we went back to our enhancer subsets. And this time we asked whether enhancers that are shared or supported by more methods have greater evidence of function using other biological features, things other than sequences. And so for this analysis, I'm going to show you results for 2 markers, one of which we've looked at before. So we'll look at enrichment for those expression-associated variants that we talked about a few slides ago, as well as for evolutionary conservation. So these would be regions of the genome that are shared across multiple species or long periods of evolutionary time. The intuition here is that things that have been kept the same over extremely long timescales are probably important and functional. And we already know from previous literature that enhancers are often conserved. So we might expect true enhancers to have higher levels of evolutionary conservation. So in this plot, in order to ask whether shared enhancers are more enriched for functional markers, we bend our enhancers by the number of methods that predict that region to have activity. So from those that are unique on the far left side of our plot to those that are highly shared on the far right. And the y-axis here is our enrichment for overlap with those expression-associated variants. When we plot the enrichment for each of these bends, we don't see very much improvement. The unique enhancers are not any, or the shared enhancers are not any more enriched than the unique enhancers for these expression-associated variants. For evolutionary conservation, we ran a similar analysis, looking at enhancers with increasing support across methods versus enrichment for the overlap with conserved elements. And here we see only a modest increase, again suggesting that taking the intersection or the shared enhancers does not provide us with a more high confidence or necessarily functional set of regions. And so despite the use of identical terminology to describe these regions in the literature, there's actually a great diversity in enhancer definitions that produce different sets of annotations. And so the consistent lack of agreement between methods demonstrates that our working definitions of enhancer actually have really low overlap. And so when interpreting non-coding variants or trying to characterize enhancers in a new biological context or tissue, we should be mindful that using a single strategy is not sufficient to catalog enhancers, especially given that differences between enhancer sets can lead to contradictory interpretations. And so given the lack of a clear gold standard and the substantial disagreement between strategies, it's not great to identify the single best method or necessarily to combine enhancer sets in hopes that we will increase evidence of function. So in the first part of the talk, as perhaps unsatisfying as it is, I discussed how we can use computational approaches, including machine learning, to highlight a technical problem in the field, defining enhancers and using them to interpret genetic variants. And so I hope this showed you that understanding the gene regulatory switches one at a time is really important. But as I mentioned at the beginning, that's only part of the story. There's still additional information that's missing. And so in the second half, or the second piece of this presentation, I'm going to tell you about some methods I'm currently developing to integrate enhancer annotations within a broader context, including information about the three-dimensional structures of DNA. And these methods will better address complexity in gene regulation than approaches that would consider enhancer elements in isolation. So it's been well established that genes can be and often are regulated by multiple enhancer sequences, as illustrated in this schematic from the very beginning of this talk. And while I'll expand on the term in a few slides, I wanted to introduce this idea of an enhancer landscape, which is essentially the set of enhancers and their genomic features that are associated with the regulation of a single gene. So in this case, the enhancer landscape of gene X would be enhancers A and B. And so we know that enhancer landscapes can play an important role in maintaining robust gene expression. An example is a study from 2018. And so this gene, GLI3 in black, is involved in limb development in the mouse. And it's regulated by two enhancers shown in green and teal in its enhancer landscape. And when the gene is expressed properly, the mouse develops normally. But if the gene is knocked out and no longer expressed, the mouse develops with a polydactyly phenotype. Interestingly, though, the enhancer landscape provides some robustness to variation that might otherwise disrupt the expression of this gene. So when only one of the enhancers, say the one in teal, is knocked out, the gene expression is still maintained and the mouse develops normally. The same if you knock out the green enhancer, the mouse still develops normally and the level of gene expression remains stable, despite the loss of one of the regulatory elements. It isn't until both of the enhancers are disrupted that gene expression is affected and the polydactyly phenotype develops again. This result is important because it suggests that considering the full enhancer landscape of a gene is necessary to understand or to predict the effects of non-coding variation in enhancer elements. The enhancers themselves are not the only important piece to understanding this landscape. And previous work has shown that enhancers act both in a sequence-specific but also a physical manner. So the genome actually loops to bring enhancers close to the genes that they target. And three-dimensional chromatin loops, like the ones shown in red and blue, form interaction domains that facilitate enhancer gene contacts and insulate other genes from being improperly activated. And so over the past few years, new technologies have developed that enable us to assay the interaction frequencies and create these or visualize these domains. And the data from these is often visualized in the form of a heat map, like the one here, where the intensity of the colors represent the frequency of interactions between any two locations in the genome. So in the schematic above, you can think about stretching out chromatin loops back into a linear sequence. And the triangle shape of this heat map shows the high level of interaction within the loops and few to no interactions outside of those loops. In this way, enhancer landscapes are also influenced by this three-dimensional chromatin architecture. And so using enhancer annotations and chromatin interaction data, I defined enhancer landscapes for all genes across 10 different human tissues. So I'll again start by orienting you to my schematic here. So the black line across the center of the screen would be the DNA strand. And we can show genes represented here by just the transcription start sites. Then we can overlay some enhancer elements, interaction domains, and individual interactions between different genomic locations within these domains. And so I define the enhancer landscape of a given gene. For example, we'll start with the one in red, as the set of enhancer elements associated with that gene by a significant chromatin interaction. And so using this gene level definition of an enhancer landscape, in the liver, 71% of genes are linked to enhancers and 68% are associated with multiple enhancers, suggesting that the idea of a large enhancer landscape is fairly widespread. And so we then evaluated whether properties of the gene's enhancer landscapes, or the number of elements associated with those genes, was associated with other gene level attributes. And so to start, we hypothesized that genes expressed in a given tissue would have a larger number of active enhancers because transcription is being regulated in that tissue. And so, for example, in the liver, the median number of enhancers for an expressed gene is 12, compared to 6 for genes not expressed in the liver. So in this box plot, you can see the difference between expressed and not expressed genes and the number of liver enhancers on the y-axis here. When considering expressed genes, we actually observe a pretty wide variability in the number of enhancers associated with each gene, or the size of that enhancer landscape. This isn't surprising, considering we would expect the number of enhancers associated with each gene to depend on more than just the level of gene expression. We might expect that tissue specificity of the gene and the activity patterns of the enhancers should also contribute. So we next hypothesized that genes with tissue-specific expression patterns would have a higher proportion of tissue-specific enhancers. And here we do find that the proportion of tissue-specific enhancers in a landscape, so that would be our x-axis in this plot, is higher for genes with tissue-specific expression, shown in blue, than it is for expressed genes overall, shown in purple. And so the example on the right is in the liver, where we compared that proportion of tissue-specific enhancers in a landscape between genes with liver-specific expression and those that were just expressed in the liver, but not specifically. And I know these plots are probably quite small, especially if you're watching this on a laptop, but I wanted to show them to try to demonstrate that this trend actually generalizes across all 10 tissue types that we considered, suggesting that this pattern of tissue-specific genes having more tissue-specific enhancers is a general pattern. And so we hypothesized then that other gene attributes could also be associated with the gene level enhancer landscape because of different levels of constraint on their expression. And so in this plot, we're going to focus again on data from the liver. And so the x-axis here is the number of liver enhancers in the enhancer landscape of a gene. And the y-axis is going to be different gene sets. So we could start with our expressed genes and our genes with tissue-specific expression that we've looked at before. I also curated 3 additional gene sets that were likely to be under different levels of constraint. So one is loss-of-function intolerant genes, or genes that are largely intolerant to mutations that would disrupt the protein. The next one is essential genes. So these are genes that are required for growth and cell proliferation across all cell types. And finally, housekeeping genes. So these are genes that are broadly expressed or expressed at a similar level across all tissues. And so we expected that genes in these different categories would have more associated enhancers because having a larger enhancer landscape provides potential for regulatory buffering, like we saw in the example with the mouse, or potentially a finer control of expression. But we instead found that housekeeping, loss-of-function intolerant, and essential genes are not strongly associated with a higher number of enhancers. And this suggests that further analysis is going to be required to understand the relationship between constraint on expression and the size of the enhancer landscape. And I just want to close with this last analysis about how the attributes of enhancer landscapes are important, and understanding the last few plots is important because these landscapes might influence the likelihood that a genetic variant would associate with disease, or a genetic variant in this landscape would associate with disease. And so after establishing that enhancer landscapes vary across genes and are associated with some gene level features, in this final analysis, we asked whether attributes of the enhancer landscape are associated with differences in enrichment for non-coding genetic variation. So this plot is going to divide enhancers into 4 categories based on their landscape attributes. First, on the left are enhancers that are active in landscapes with lots of enhancers, more than 5, and looking at whether those enhancers have conserved or not conserved activity across species. And for these, we observe a slight depletion for expression-associated variants, like we talked about earlier in the presentation. On the right are enhancers that are active in landscapes with few enhancers, less than 5, and again, whether those enhancers are conserved or not conserved across species. Here we observe a slightly larger depletion for expression-associated variance, but none of the depletions in this plot are statistically significant. When considering a similar plot, comparing enrichment for trait-associated variance, or things associated with relevant diseases, we observe that there's enrichment and enhancers with conserved activity in these landscapes that are very large. This suggests that while some large enhancer landscapes may contain redundant elements and buffer effects of genetic variation, probably not all of the elements are redundant, and so variation in these regions can still be associated with disease. So moving forward, it's going to be really important to develop improved methods to further describe the attributes of these gene regulatory landscapes, to understand which elements might be redundant, and understand how the attributes of regulatory landscapes modulate genetic variant effect. And so just to close here, by leveraging experimental data across 10 different human tissues, we defined and highlighted features of enhancer landscapes. I talked about on the gene level here, but we actually did this in 2 different ways that I didn't have time to talk about today. We also showed that the size and the tissue specificity of the landscape is associated with tissue specificity of the gene. And this is useful because these enhancer landscapes may provide information that helps us interpret the effects of non-coding variation. And so, in summary, this work highlights some key limitations in our understanding of gene regulation and defines a model to help better understand the framework of gene regulatory architecture, or these enhancer landscapes. But in the future, we still need new methods, some to integrate new experimental data, identify potential regulatory regions, and better understand the similarities and differences between predictions that I talked about at the very beginning. In addition, I'm hoping to continue to build on these frameworks to quantify redundancy in gene regulatory landscapes, and ultimately to help improve the prioritization of non-coding genetic variants, or better predict which ones might be involved in disease by including enhancer landscape features into these models. And with that, I'd like to thank my collaborators and some of the funding for this project, and you for your attention. I'm happy to take questions if we have any time. So thank you all.
Thank you so much, Dr. Benton. Does anyone have any questions? Well, I have one. It's about your undergraduate research. So I know you went to Baylor. Were you involved in undergraduate research at Baylor?
So I actually wasn't involved in research at Baylor. I did an internship in my undergrad working at the University of Tennessee and the Oak Ridge National Lab. They have one of the biggest supercomputers in the world. And so I worked with scientists there to develop some parallel computing resources. If anyone's used BLAST in any of their genetics classes, we were developing a version of BLAST that could run on massive supercomputers. But undergraduate research is great. That's what actually got me interested in grad school in the first place. I was thinking about going to med school, and after my undergrad research experience, I realized that that was what I wanted to do instead. So I can highly recommend.
I have a quick question on the first part of your presentation on like the Jaccard similarity and then your machine learning models.
Sure.
So for the Jaccard similarity, I noticed you just use like the straight values. Mm-hmm. And I know that with Jaccard, there's like a limitation with like the significance of like what the value you get is, right? So it's like if you have 2 sets of like giant sizes, then the overlap may or may not be significant. So how did you address that limitation in your research?
Yeah, that's an excellent question. So that's definitely a limitation. And the other limitation with that Jaccard similarity is that if you have one really big set and one really small set, the really small set might be a perfect subset of the bigger one, but the maximum amount of similarity is pretty low. So the plots I showed you today are that straight similarity calculation, but we also developed a relative, what we called a relative Jaccard similarity. And so that was the Jaccard similarity normalized by the maximum possible similarity for the comparison between those two sets. So if one was really big and the other was very small, the maximum amount of similarity would be low. So it was more like a measurement of how close to the maximum similarity you achieved. Those values were also very low, generally below 0.5. So not quite as dire as the straight values, but still not as high as we would expect.
Awesome, thank you. And then I guess I had a follow-up question on your machine learning model. So I think you said that you used the Jaccard for like determining your statistical significance for it. And so I guess I just wanted to ask, like, why did you choose Jaccard over like other similarity measures such as like cosine similarity, stuff like that?
Yeah, so we used the Jaccard similarity primarily to divide up the enhancers between those that were unique and those that were highly shared. For the features of our machine learning model, we actually just used the frequency of different K-Mer sequences across the length of that enhancer. So we use the Jaccard similarity, or the overlap between enhancers, to kind of divide our sets into two classes, positives and negatives, but then the features themselves were actually just the frequency of sequences of short sequence motifs across the length of the enhancer. Does that help?
Yeah, thank you.
I don't think there are any further questions. But if you guys think of anything, feel free to email Dr. Benton. I'm sure she'd be happy to get back to you.
Definitely. I'm putting my email in the chat so that you have it. I'm also fairly easy to find on the website, but all right. Well, thank you all again for your attention and the questions. It was great to talk to you.
Thank you so much. All right, guys, don't forget to connect with us on our Twitter, our Instagram, and our website, and lately our YouTube, because we've been posting all of our general meetings on YouTube. We'll also be posting the events from Research and Internship Day on YouTube, so definitely check it out if you haven't. And then thank you guys for coming. Go ahead and take a picture of this QR code and fill out the form so that you can get your one general event point. And then stick around for the social that's happening right now. We're going to be playing Among Us. But thank you guys so much for coming.